喺呢度我就要話俾你聽，原來考評局係真係會重用題目㗎。2020年嘅第5條咧就有關於蒸騰作用同埋科學探究嘅。咁呢一幅圖咧就顯示咗一個去比較翻一個盆栽嘅葉片嘅上表皮同埋下表皮嘅蒸騰速率嘅實驗裝置嚟嘅。咁呢個裝置咧包括咗上面同埋下面一個相同嘅中型罩啦，個 bell jar 啦。咁啊兩個中型罩之間咧就夾住咗呢棵植物嘅一塊葉嘅。咁呢個罩入面咧，亦都放置咗個化學品 X 啦，咁去吸收翻啲水氣。咁成個裝置咧，由呢個 U 型管啦，去到呢個中型罩啦，去到另一邊嘅 U 型管咧，都係密不透氣嘅。咁喺呢個實驗嘅開始之前啦，同埋5小時之後啦，咁我哋都會量度翻喺呢兩個中型罩入面嘅化學品 X 啦，佢哋嘅質量嘅。咁所以你見得到啦，喺 M 呢個中型罩咧，佢哋個化學品 X 咧就由 2.4 gram 去到 2.49 gram， 而喺 N 嘅呢個中型罩咧，佢呢個化學品 X 咧就由 2.43 gram 就去到 2.56 gram 啦。我哋就先睇下 Part A 同埋 Part B 咯。咁 Part A 咧就問我哋啦，呢一個實驗裝置利用咗以下邊一個嘅參數去量度翻植物嘅蒸騰速率咧？咁啊喺翻呢個格格度剔翻啱嘅答案啦。而第二部分啦，就係問翻呢個實驗裝置係點樣量度呢個 A1 嘅參數啦。Part B 就要我哋列出翻喺呢個科學探究入面係受到實驗裝置所控制嘅兩個變項，亦即係問緊我哋控制變項啦，係咪？約略講完 A、B 嘅題目啦，其實佢考緊我哋啲乜嘢呢？第一啦，就係考翻我哋對於變項嘅理解啦。咁我哋有獨立變項啦、因變項啦，同埋控制變項啦。咁之前我哋都拍過一段片咧，解釋翻啦。IV 咧就係我哋去改變嘅 ，DV 咧就係我哋去量度嘅 ，CV 咧就喺整個實驗裝置或者不同嘅實驗裝置之間咧都係保持不變嘅。第二樣要考下我哋啦，就係整個裝置咧係點樣運作嘅。咁頭先我哋約略都講過噶啦，其實就係睇翻呢個化學品 X 咧，佢哋嗰個重量嘅改變。而點解佢會有個重量改變呢？其實就係因為佢哋會吸收水氣嘅。而去到第三咧，正正就係題目 Part A1 咧問我哋嘅嘢啦。究竟我哋點樣去量度呢個參數啦？咁我哋先睇下 Part A1 咯。下面邊一個參數去幫我哋量度翻蒸騰嘅速率呢？咁究竟咩叫蒸騰啊？就係失水啊嘛。咁所以啦，我哋量度緊嘅咧，其實就係個葉片嘅失水量啦。我哋整個裝置啦，又如何去量度翻葉片嘅失水量呢？就係透過翻化學品 X 啦。咁啊，因為喺個葉嘅表面呢，我哋有個水氣嘅蒸發，咁呢個就係蒸騰作用啦。而呢個化學品 X 咧，當佢吸收咗水氣之後呢，佢嗰個質量就會上升。咁所以你見到啦，2.4去到2.49，2.43去到2.56啦。咁呢個咧正正就係我哋嘅 DV。咁所以啦，有少少概念咧要釐清翻啦。整個實驗嘅 DV 究竟啦係葉片嘅失水量啊，定係化學品 X 嘅質量改變呢？其實係化學品 X 嘅質量改變，呢個先係我哋真正量度緊嘅變化緊嘅嘢。因為呢個先係符合翻 DV 嘅定義，就係隨住自變項而改變嘅變項，亦即係喺個實驗當中要量度嘅變項。你而家量度緊乜嘢啊？就係量度緊化學品 X 嘅質量改變啊嘛。而呢個化學品 X 嘅質量改變呢，就反映咗葉嘅上表皮同埋下表皮嘅蒸騰速率啦。跟住啦就去到 Part B 啦。咁究竟喺整個實驗當中有啲咩嘅控制變項呢？第一個啦就係植物嘅種類啦，同埋葉嘅種類啦。你就冇得咧葉面嘅上表皮咧就用 A 嘅植物，而葉嘅下表皮咧就用 B 嘅植物。咁如果 A 同 B 兩款植物根本就唔同種類嘅，其實係唔公平嘅。第二啦就係表皮嘅表面面積啦，亦即係呢個中型罩所覆蓋嘅圓形表面咧，佢哋嘅面積應該要一樣嘅。如果啦，你葉嘅上表皮用嘅中型罩大啲，而葉嘅下表皮用嘅中型罩就細啲，咁其實都係唔公平嘅。第三啦，就係整棵植物所承受嘅風速，又或者上表皮同下表皮所承受嘅風速啦。而啦，你就將整棵嘅植物啦，呢個實驗裝置啦，就擺喺個實驗室入面啦。 你就唔能夠咧喺個葉嘅上表皮咧就攞把細嘅風扇嗰啲芭蕉扇咧不斷咁吹佢，但係葉嘅下表皮咧就唔吹佢，咁其實都係唔公平嘅。咁啊，因為風速咧係會影響咗蒸騰速率噶嘛，係咪？第三啦，就係含有水氣嘅空氣體積啦，即係咧要用相同嘅中型罩咧去覆蓋啦，亦即係咧你又唔能夠上面用一個大啲嘅中型罩，下面咧就用細啲嘅中型罩。咁啊，因為啦個中型罩大啲嘅話咧，能夠保存住多啲嘅水氣，亦即係話喺呢個中型罩入面嘅濕度咧，其實係增加咗嘅。我唔理你呢個化學品 X 吸咗幾多水氣。咁由於呢度佢大啲啊嘛，咁即係話佢有機會咧係儲存咗多啲嘅水氣，都係會令到個實驗咧唔準確同埋唔公平嘅。而去到下一條之前咧，有一樣嘢咧，我想提下大家，都係有關於實驗裝置嘅。今次題目冇問嘅，下次可能問你啦。題目就講到明俾你聽㗎啦。呢個實驗裝置咧係密不透氣嘅。其實啦，下一次嘅題型變奏咧，佢就會問下你啦。其實題型變奏咧，下次佢就可以問下你啦。喺整個實驗裝置嘅 U 型管啊，究竟有咩嘅作用呢？入面裝住啲油又有咩意思呢？就係唔俾啲水氣喺個實驗外面嘅地方進入去呢個中型罩入面啊。咁你會問啦，咦？咁我唔擺油擺水得唔得啦？擺水你咪豬咗咯，因為呢度係水嘅話，啲水係會蒸發噶嘛，蒸發咗入咗個中型罩嘅話啦，咪會俾個化學品 X 吸收咗去咯，即係無端白事啦。即使你嗰個葉嘅上表皮或者下表皮，佢失水可能本身係好少嘅，但係你呢個 U 型管入面，你無情情擺咗啲水響樹，啲水喺度蒸發咗上去，咁會令到個化學品 X 咧吸多咗水啦。都係令到個實驗咧變得唔準確，係有個錯誤響樹嘅。好，跟住啦就去到 Part C 啦噃。第一部分咧就要我哋比較翻中型罩 M 同埋中型罩 N 入面化學品 X 嘅質量改變，從而啦又得出咩嘅結論呢？今次呢一部分咧就考翻我哋嘅全視技能啦，就係計數咯，同埋得出結論啦。咁尤其是係得出結論啦，你就要用翻直線抽擊答題法啦。咁整個答題應假啦，題目就講到明噶啦，要我哋比較翻化學品 X 嘅質量改變啊嘛。咁喺呢一句說話入面有冇講到質量改變俾你聽？係冇嘅喎，所以啦要計一計數啦。兩者咧其實都係上升嘅，不過咧喺中型罩 M 咧嘅化學品 X 咧就上升咗 0.09 gram， 而喺中型罩 N 咧佢嘅化學品 X 咧就上升咗 0.13 gram。咁所以啦，第一個比較咧你就會發現啦，其實大家都升。不過啦，喺中型罩 N 入面嘅化學品 X 咧，佢就相比起中型罩 M 嘅化學品 X 咧，再增加咗嘅質量咧係大一啲嘅。咁啊，橫掂咪計得數啦，不妨將你嘅答案咧擺埋入去咧，就令到個答案咧更加完整啦。而第二部分啦，就要得出結論咯，直線抽擊。整個題目就係想去比較翻葉嘅上表皮同埋下表皮嘅蒸騰速率。呢個葉底嘅部分中型罩 N 咧，佢個化學品 X 係吸多咗水，咁自不然啦，就係葉嘅下表皮嘅部分，佢個失水速率咧就會比葉嘅上表皮嘅部分咧為之高一啲啦。然後啦，根據呢個結論咧，Part 2就問我哋有冇一個可能嘅解釋啊？咁所以題目咧就考翻我哋啦，要提出一個假說去解釋某一啲嘅現象啦。今次就係解釋翻 Part C1 嘅結論啦，就係葉底失水嘅速率係比起葉面嘅失水速率為之快嘅。咁呢一部分咧就係考翻我哋有關於蒸騰作用嘅機制啦。第一個方法就係啲水氣就透過氣孔咧就擴散去到大氣當中，而第二個失水嘅途徑咧就係啲水係會透過葉嘅角質層咧去流失於大氣當中。所以啦，喺我哋嘅題目當中咧，我哋就會發現有唔同類型嘅假說啦，例如啦，氣孔嘅多少啦，氣孔嘅密度啦，氣孔嘅結構，即係話佢開大啲定係開細啲啦，又或者係個角質層嘅厚度啦。但係個題目係要你提出個假說去解釋翻 C1part 嘅結論啊嘛。C1part 嘅結論就係講緊葉面同埋葉底嘅失水速率係有唔同嘅。所以喺呢一 part 我想提翻大家嘅就係記緊喺你呢個假說入面係要提及翻葉面同埋葉底嘅結構。所以你嘅答案就唔能夠淨係講翻氣孔嘅數目，而係要講緊上表皮同埋下表皮嘅氣孔數目有唔同。 上表皮同下表皮嘅氣孔密度有唔同，上表皮同下表皮嘅氣孔構造有唔同，同埋上表皮同下表皮嘅角質層嘅厚度有唔同。呢個先係一個完整嘅假說。啦，就到第三部分啦。近呢幾年呢，係多咗呢啲題目嘅。呢一條題目呢，就係2020年嘅。咁啊，因為啦撞正呢，就係 COVID-19， 大家係少咗機會做實驗噶嘛。所以啦喺個題目當中呢，佢就考翻大家多啲做實驗嘅技巧啦。例如去設計一個實驗咯。咁 Part 3咧就問翻啦，試提出一個可行嘅方法去測試翻頭先喺 C2 part 所提出嘅假說。咁頭先 C2 part 我哋就講啦，葉面葉底佢哋嘅氣孔數量啊、氣孔密度啊、氣孔結構啊，同埋角質層嘅厚薄嘅。咁而家個題目咧就考翻我哋科學探究嘅技巧咯。有冇一啲裝置你係能夠建立得到出嚟㗎？去測試翻唔同類型嘅假說啦。咁當中咧，我哋要提及翻個 DV 啦。今次個 DV 咧就係根據翻我哋嘅假說嘅氣孔嘅多少啦、氣孔嘅大細啦、角質層嘅厚薄啦。而最重要嘅咧就係你肯定你呢個裝置係能夠幫你比較到葉面同埋葉底嘅結構嘅。咁同埋啦，你就要揀翻一個適當嘅器具去幫你做一個量度啦。第一類型嘅裝置去探究翻個氣孔多少嘅咧，究竟我哋用咩辦法呢？我哋有啲適當嘅工具咯，例如我哋要用翻顯微鏡啦，我哋要用指甲油去搽喺個葉面嗰處，跟住搣出嚟啦，獲得一個表皮嘅倒模去做翻顯微鏡嘅觀察啦，或者啦，將塊葉擺喺個熱水當中，然後啦，我哋就要去數下啦。今次我哋要觀察嘅係乜嘢啊？例如啦，觀察翻氣孔嘅數量啦，又或者啦，當塊葉120，又或者我哋將塊葉浸落熱水嘅時候呢，我哋就要數翻喺個葉嘅表面，佢能夠釋放咗幾多個泡泡氣泡啦。但係最緊要嘅就係你要比較翻葉面同葉底喎。咁所以整個答案呢，你會發現啦，有紫色嘅部分就係用咩器具啦，紅色嘅部分究竟我哋觀察緊啲乜嘢啦，同埋藍色嘅部分究竟我哋係咪真係比較緊葉面同葉底啦。第二類型嘅裝置呢，就係想去研究下氣孔嘅大細嘅。咁我哋仍然啦，都係用翻一個適當嘅器具啦，就係顯微鏡啦。然後我哋就比較翻個氣孔嘅大細，而記緊你係要比較翻葉面同葉底嘅氣孔大細。而第三類型嘅比較呢，就係角質層嘅厚薄啦。咁首先啦，我哋要獲得塊葉嘅切面啦。首先啦，我哋要獲得塊葉嘅切面啦 ，cut a section of a leaf， 然後又係用顯微鏡去觀察咯。今次我哋要觀察嘅呢，就係角質層嘅厚薄，又或者氣孔嘅結構，究竟開大定係開細啦。而去到最尾又要提及翻，究竟你係咪比較緊葉面同葉底啊？所以啦，喺呢一 part 咧，大家要留意翻啦。當你做實驗係咪有器具㗎？當你做實驗係咪有觀察㗎？而當你做實驗嘅時候啦，係咪有機會要比較㗎？係呢三個問題你一定要解決㗎啦。當你做實驗嘅時候咧，呢三個問題記住問一問自己咯。過往咧我都拍咗一啲片係有關於實驗嘅假說啦，同埋啦唔單止由2020開始嘅，其實過往咧間唔時都會問下大家咧去設計啲實驗，或者呢個實驗嘅裝置究竟做嚟為乜嘅？例如啦，當初咧我哋過氧化氫酶嘅呢個實驗咧，正正就係決戰 SBA 啦，正正就係要你設計實驗啦。咁啊，快啲睇翻段片溫下書咯。跟住啦，又去到一點出發啦。今次嘅題目咧就由蒸騰速率咧去問起嘅。今次嘅題目咧主要就集中翻葉嘅上表皮同埋下表皮啦。而另一款影響住蒸騰速率嘅因素咧就係環境因素啦。咁上表皮同下表皮啦，氣孔同埋角質層，正正係呢條題目考我哋嘅嘢啦。當中我想提大家嘅呢，就係有關於唔同植物嘅適應方法啦。陸生嘅植物、浸沒嘅植物同埋漂浮嘅植物，究竟佢哋個葉面葉底，佢哋嘅氣孔啊、角質層啊，有冇啲咩嘅唔同呢？例如啦，漂浮植物好似嗰啲荷花咁嘅款嘅，喺個葉底呢，原來冇角質層嘅喎。咁冇角質層嘅話啦，又有冇氣孔呢？原來佢哋嘅葉底係冇氣孔嘅，而佢哋嘅葉面呢係多一啲氣孔嘅。 如果成棵植物浸咗落水嘅話啦，咁佢又有冇角質層呢？又有冇氣孔呢？咁過往咧我都拍咗一段片咧，去講翻植物嘅進化史嘅。咁大家快啲咧就去翻留言區咧，去睇翻段片，溫一溫書咯。然後啦就去到環境因素啦，我哋有風速啦、相對濕度啦、溫度啦同埋光度嘅。咁啊有少少提示啦，呢三個環境因素咧都係直接影響住蒸騰速率嘅因素嚟嘅。而光度咧就係間接嘅影響嚟嘅啫，因為光度咧就只係影響住嗰個氣孔開大定開細，然後先再影響翻個水氣嘅蒸騰速率。但係溫度、濕度同埋風速咧係直接影響住個水嘅蒸發速率啦，同埋擴散嘅速率嘅。咁呢一 part 咧大家又要留意咯。然後啦，整個實驗就係問科學探究啊嘛，3個 V 分清楚未呢？而個實驗亦都有問我哋有關於假說嘅問題，亦都有叫1、2、3，亦都叫我哋去設計嘅實驗啦。過往咧都有唔少有關於蒸騰速率嘅 MC 嘅，尤其是係2018年呢2條題目。如果我同你講考評嗰班人係真係會翻兜題目嘅，你信唔信啊？我題目都輯埋俾你哋睇啦，連啲數字都一樣㗎，係咪黐線㗎？而個題目都係一樣㗎，問你化學品 X 點解佢個質量會改變，而根據佢嗰個化學品 X 嘅質量改變，究竟你會得出咩嘅結論？正正喺 Part A、Part B、Part C 都有問你嘅題目，將 MC 變成長題都有可能啦。難保有一日會將長題變做 MC， 就更加有可能啦。咁多位，所以啦，咁多位兄弟姊妹，你要去應付考試，其實真係做下卷啦，做下練習啊，係一定有你着數嘅。你做咗練習，你學多咗嘢，一來係你自己嘅。如果你諗下當初你喺18年操掂呢2條 MC，去到2020年呢條 long question 2條題目，你實食冇黐牙啦。咁多位。2020 question 5 is about transpiration and scientific investigation. The diagram shows an experimental setup used to compare the transpiration rates of the upper epidermis and lower epidermis of a leaf of a potted plant. The setup consists of 2 identical bell jar M and bell jar N, placed one above the other with the leaf of a potted plant in between. Chemical X was placed into the jar, chemical X, and then to absorb the water vapor. And the whole setup was made airtight. That means the air outside the setup, especially the water vapor, they cannot enter the setup. And the masses of the chemical X in the jar were measured at the beginning and after 5 hours. You can see the result. Chemical X in the bell jar M, initial mass 2.4 grams and final mass 2.49 grams. And for the chemical X in bell jar N, initial mass 2.43 grams and the final mass is 2.56 grams. So let's take a look at the questions first. Part A1. Which of the following parameters is used to measure the transpiration rate of the plants in the investigation? Part A2. How can the above setup measure the parameter chosen in Part A1? Part B. List 2 variables which have been controlled by this setup during the investigation. Part A and Part B actually it's checking us the concept about the variables in the scientific investigation. So you can watch the video again to get familiar with these 3 variables. Independent variable, the factor we are investigating, we are studying. Dependent variable, the variable to be measured. Control variable, the variable that are kept the same or are constant in different setups. And then we need to understand how does the setup works. The bell jars it trap a certain area of the leaf surface, and when the leaf surface lose water, the water vapor will be absorbed by the chemical X. And then finally determine how to measure the parameter. So let's take a look at part A1, because we are going to measure the transpiration rate of the plant. So what is transpiration? It's talking about the water loss. For the answer must be the amount of water lost by the leaf. And then for part A2, how can the above setup measure the parameter? The water vapor is lost from the leaf surface through transpiration, and the mass of the chemical X increases after absorbing the water vapor. So one thing I would like to mention is that what is the dV of this experiment? It is the changes of the mass of the chemical X because that's the variable we are going to measure. This parameter, it will reflect the transpiration rate of the upper epidermis and the lower epidermis of the leaf. And then for part B, we need to list the two variables which have been controlled by the setup during the investigation. Anything we need to keep constant in the whole setup. For example, the type of the plants or leaves. So for example, for the upper epidermis, we use the plant A. And then the lower epidermis, we use the plant B. So plant A and plant B, they are the different plants or different species of the plant. Is not fair. So that's why we need to use the same pot or same leaves being investigated. And then the surface area of the epidermis, that means the identical vase. We need to use the same bell jar to cover the same area of the leaf surface. And then for the wind speed, we need to put the whole setup in the enclosed system. For example, we put it in the laboratory. We need to ensure that the air current or the wind speed on the upper epidermis, it should be the same as the lower epidermis. We cannot use a little fan to blow the air on the upper epidermis because the higher wind speed, it will increase the transpiration rate. Finally, the volume of air containing the water vapor confined by the identical vase. So for the bell jar M and bell jar N, so what if we use a bigger bell jar M? So actually, the bigger bell jar M, it may trap more water vapor. And then the relative humidity in the bell jar M will be higher than that of the smaller bell jar N. Is also not fair. Therefore, you can tell us that we use the same pot, same leaf, covered by the identical vase, enclosed system, or confined by the identical vase. Before we move on to part C, I would like to talk about the possible question variation. In this question, it doesn't ask you this thing, but maybe next time it will ask you what is the function of putting oil in the U-shaped tube. So you can see there are two U-shaped tubes in the setup. And actually, the hint is that the whole setup was made airtight. So apart from the junction of the bell jar M and N, the other important part of the setup is the U-shaped tube. Why do we put the oil inside? Because it will block the water vapor coming from outside to the bell jar. Therefore, the water vapor appear in this bell jar. It should come from, so you can imagine that the water vapor in this bell jar, they should come from the leaf surface by transpiration. So you may ask that, can I add the water in the U-shaped tube? No. If you really put water in the U-shaped tube, the water, it will evaporate. And there will be more water vapor go into the bell jar N or M. So it will affect the result of the experiment and make it inaccurate, and even it is a mistake, not an error at all. And then we go to part C. Part C1, compare the changes in the mass of the chemical X in bell jars M and N, and what conclusion can you draw? This question checks you the cyanotic skills, calculation, and draw conclusion. Especially, you need to use the straight to the point method to draw the conclusion. The scaffolding is that, firstly, you need to take a look at the question again. Actually, it doesn't tell you the changes in the mass of the chemical X. So that's why you need to calculate the change for the answer. Chemical X in the bell jar M increases for 0.09 gram, and for chemical X in bell jar N, it increases by 0.13 gram. So we need to calculate and compare the changes in the mass of the chemical X in bell jars M and N. The increase in the mass of the chemical X in bell jar N is greater than that in the bell jar M. And then we need to draw the conclusion directly based on the result. We can recall the question. We are going to compare the transpiration rates of the upper and lower epidermis of the leaf of a potted plant. Therefore, this result, it shows us that the rate of the water loss from the lower epidermis is higher than that of the upper epidermis. Very direct conclusion. And then for part 2, we need to suggest one possible explanation for this conclusion. Test us the skills to propose a hypothesis to explain the conclusion. So the concept checking is that we need to recall the mechanism of transpiration. The water can diffuse through the stomata into the atmosphere, or less than 10% of the total water loss takes place through the cuticle of the surface of the leaf. So the possible explanation, it could be related to the number of stomata, stomata density, stomatal structures, it open bigger or open smaller, and the thickness of the cuticle. And one reminder for this question is that because we are going to propose an explanation for the conclusion from part C1. And for the part C1, we are comparing the rate of water loss from the lower epidermis and the upper epidermis. Therefore, you need to make sure that the possible explanation is also related to the comparison of the upper and lower epidermis of the leaf. Therefore, the whole answer, it should be the different number of stomata of upper and lower epidermis, the different stomatal density of the upper and lower epidermis, different stomatal structures of the upper and lower epidermis, and different thickness of the cuticle of the upper and lower epidermis. And for the part C3, it is a common question type for the SBA question, especially within these 2 to 3 years. Due to the COVID, we have less time for the SBA, for the experiment. There will be more questions to ask you to design the experiment, design the investigation. So we need to propose one workable method to test the hypothesis in part C2. And this question is checking us the skills about the scientific investigation to establish, to design a setup to test the hypothesis. And then we need to record the DV based on the hypothesis. We are comparing the number of stomata, the stomatal density, the size of the stomata, and the thickness of the cuticle. And the most important thing is that you need to make sure you are really comparing the upper and the lower epidermis of the leaf. Just like what we do in the hypothesis, we are comparing upper and lower epidermis of the leaf, right? Finally, we need to choose the suitable apparatus for the measurement. So for the first type of design, how can we count the number of the stomata of the leaf surface? So we can use the microscopic examination or use the nail polish to obtain a mold of the epidermis for the microscopic examination, or we immerse the leaf in a hot water. And then what do? We count the number of the stomata per unit area, or we count the number of the bubbles appearing on each surface per unit area if we put it in the hot water. And remember that you need to compare them. Therefore, you need to mention the upper and lower epidermis. So you can see the purple part is the suitable apparatus. The red part is about what do we do, and then the blue part is about the comparison. So you see that in the scientific investigation, we need to choose the suitable apparatus, and then we also need to know that what are we going to do, and then we also do the comparison in the investigation. Therefore, when you are doing the design of the setup, therefore when you are doing the design of the experiment, that you need to recall these 3 factors. Secondly, we talk about the size of the stomata. So we still use the microscopic examination of the epidermal pier, and then we compare the size of the stomata on upper and lower epidermis. And finally, we compare the stomatal structure or the thickness of the cuticle. Firstly, we cut a section of the leaf for the microscopic examination, and then we can compare the appearance of the stomata or the thickness of the cuticle of the upper and lower epidermis, the purple, the red, and the blue part. And before that, I made some video about the hypothesis, and there were different questions about the design of the setup, especially for the 215 question about the catalyst investigation. So let's talk about the curriculum mapping. This question starts from the transpiration rate, and it is controlled by the upper and the lower epidermis and also the environmental factors. So in this question, when we talk about the epidermis, we mention the stomata and the cuticle. And one thing I would like to mention is that different types of the plants, they have different types of adaptation. For the land plants, submerged plants, and the floating plants, do they have the cuticle? Do they have the stomata? For example, for the floating plant, just like the lily, so the lower epidermis, it is immersed in the water. Therefore, the lower epidermis of the floating plant, they do not have the cuticle, and then they can absorb the water directly, and then they do the gas exchange throughout the leaf surface. Therefore, they do not have the stomata. For the upper epidermis of the leaf of the floating plants, they have a lot of stomata for gas exchange because there is cuticle on the upper epidermis. What about for the submerged plants? The whole plant is submerged in the water. So do they have the cuticle? Do they have the stomata? That's something you need to recall the idea from the video I made before, because I make a video about the plant evolution. So I will put the link in the comment section. Please watch it as soon as possible. And then for the environmental factor, we have the airflow, air movement, that means the wind speed, relative humidity, temperature, and the light intensity. And one thing I would like to mention is that for the wind speed, relative humidity, and the temperature, they are the direct factor to affect the rate of evaporation and the rate of diffusion, and it will directly affect the rate of transpiration. However, for the light intensity, it is the indirect factor because it will just affect the degree of stomatal opening, or it will affect the temperature. So it will not affect the rate of evaporation or the rate of diffusion, or it is the indirect factor. Finally, it is the scientific investigation question. Therefore, you need to recall the three variables. Are you familiar with them? Then you need to propose the possible explanation, the hypothesis of a certain conclusion or a phenomenon. And then you need to design the experiment to test this hypothesis. And in the past, there are different questions about the transpiration, and especially 2018 question 30 and 31 in paper 1A, the MC question. If I tell you that the HKDSE, they really repeat or reuse the question, do you believe that? NANI? I even captured the question for you. You can see the setup exactly the same. Even the description of the question also the same. Come on. Even the data 2.40, 2.49, 2.43, 2.56. Unbelievable. Ridiculous. And then you take a look at the question 30 and 31. Change in the mass of the chemical X was mainly caused by, and then which of the following conclusion can be drawn from the result? These are the questions for the part A, part B, and part C of this long question. So you can imagine that they can change the MC question into long question. It's also possible to make the long question back to the MC question. Ladies and gentlemen, I really need to tell you that doing the past paper for practicing is really crucial.